everyone. Um, Good afternoon. Welcome to attend LUMICKS webinar. My name is Jason. I'm the moderator for today's session, and I am an application scientist at the Boston office. We also have Dr. Nastron Hadiat, our application scientist, joined from the California office as our panelist for the later Q&A section. Thank you all for making the effort to attend, especially under the current unusual circumstances. We are happy to have you with us. The topic of today's seminar is Decipher, the activity and mechanical properties of cytoskeletal filaments, their modes, and their role in cellular mechanobiology. Without further ado, I would like to introduce you, our speaker today, my colleague, Dr. Shima Ramasaraj. Shima received her PhD degree in physics from University of Massachusetts Amherst, where she used single molecule thread technique to study RNA dynamics. After finishing her PhD training, she joined Dr. Michael Prius and Dr. David Washer's research group at the University of Vermont as a postdoctoral research fellow. She began her research using super resolution microscopy to study localization of myosin binding protein C in cardiac muscle. A couple months ago, LUMICKS was fortunate to acquire her talent, and she began as an application scientist in our Boston office. So now I'll hand over the mic to Shima, and she will start her presentation.
Thank you, Jason, for the introduction. We are very excited to be here with you today, and we are going to talk about the technology that LUMICKS has developed to study biological processes at the single molecular level. And we are going to emphasize on applications of this technology in studying cytoskeletal filaments and their motors. So I would like to start with why single molecule studies are important. In this animation that you can see inside the cell, that I'm sure many of you have seen it, you can see a motor protein that is working on a cytoskeletal filament while carrying a cargo. And you see that processes that happen inside the cell is very complex, and cell is a very dynamic environment. Now, in order to understand these biological processes that happens inside the cell, structural biology gives us a static structure of biomolecules with the atomic resolution. But it doesn't give us the dynamic information. On the other hand, like bulk assays, like biochemical assays, gives us an ensemble average of molecular properties, and they don't give us information about the underlying dynamics of these molecules. But with single molecule studies, we can get information about subpopulations and also, for example, transient events. Different single molecule techniques have been developed over the years, and the technology that we are introducing to you today is called C-trap. And what makes it powerful is that it's a combination of optical trapping and fluorescence microscopy. So at the same time that you can apply force and measure forces of biomolecules, you can also fluorescently label them and look at those processes. And this system also has a microfluidics system with a flow chamber that works with laminar flow. And this makes even very complex assays very easy to assemble. C-trap has also an automated software which is super user-friendly, and it makes running different components of this system very easy. C-trap can be used to capture dynamics of biomolecules, as you can see in this very beautiful movie that shows two beads that are trapped using the IR laser, and the double-stranded DNA is tethered between these two beads, and you can see the binding of fluorescent proteins to this DNA and their dynamics. So here we're showing a double-stranded DNA, but this can be any other filament. It can be actin or microtubules, for example. So we use a highly focused laser light to trap objects in optical trapping. But how can we use light to trap objects? When you have a bead, for example, in the path of the light, when light gets in and out of the object because of the refraction, the direction of the light changes, and this makes a change in the momentum of the light. And because of the conservation of momentum, the bead also feels the same amount of momentum in the opposite direction. And this is the principle that is used in optical trapping. Now, when we have a focused laser light, because the intensity of the light at the center of this focused light is higher than its surrounding, the overall momentum that this bead feels wants to push it back to the center of the focus. So that's why it feels a net force toward the center. And this is how we can trap objects. Now, how is optical trapping used to measure forces or small distances? So optical trap works like an optical spring, meaning that when we have an object at the center of this focused laser light, when the bead is displaced from the center of the focus light, it feels a restoring force that wants to push it back to the center of the trap. And this force is proportional to the displacement of this object from the center of the focus light, exactly like a spring. Now, for example, we can measure the displacement of the bead using a position-sensitive detector. And once we have the displacement, by calibrating this trap to get a value for its stiffness, we can measure force. So in this way, optical trapping can be used to measure very small forces and small displacements. And we're talking here about like piconewton forces and nanometer displacements. Or, for example, optical tweezers can be used as a manipulating device to, for example, move the objects in different directions. So C-trap also has imaging, like different imaging modalities. So optical tweezers can be combined with confocal microscopy, and if you need better resolution, you can upgrade the system to STED. And for the assays that needs to be done on the surface, optical trapping can be combined with our surface assay kit, which is composed of TERF, wide field microscopy, and IRM. I will get back to this part later. For now, we want to talk about confocal microscopy. So in confocal microscopy, a focused laser light is used to excite the sample, and only the light that is coming from this small volume of sample will be detected. So this is a very nice way of getting rid of all the fluorescence that is out of this focused area and improve the signal to noise of the image. Now, by scanning the sample, we can create a 2D image of sample, or for example, we can choose to do just a line scan to create a chymograph and look at, for example, the kinetics of interactions. Or for example, you can choose to do a point scan and look at the photon counts that are coming from that like point that you're scanning. For example, here this has been used to look at the photobleaching step of this protein. So this is a very nice video that shows the workflow of C-trap. As I said, C-trap has a microfluidic system, which you can see here with the flow cell that works with the laminar flow, and it makes experiments much more easier. This is the design for the flow cell, and depending on your assay, you can have a different number of chambers, and you can flow in the different components of your assays in these chambers. For example, in the first chamber, we're flowing in the beads, and we turn on the IR laser to trap these beads that are functionalized with streptavidin. In the second channel, we have the DNA flowing. The DNA has biotin on it. So now the DNA binds to these beads, and to make sure that the DNA is tethered between these two beads, we apply force to it and extend the DNA. And now that we have the DNA tethered between the two beads, we can move to the third channel where there is a protein A, for example, fluorescently labeled, and then we go to the fourth channel with a protein B, which is fluorescently labeled. And now we can move to the buffer channel. In this way, we make sure that there is no fluorescence from like proteins that are flowing in, and it improves our signal to noise. And now we can do the confocal scanning, for example, to get a 2D image of the sample, or a chymograph, for example, to look at the kinetics. And at the same time that we're doing the confocal imaging, we can apply force to our DNA, for example, to see the effect of force. So this is a very nice way of combining force measurements and fluorescence measurements to, at the same time that you apply tension, you can visualize your biological process. Now, what applications does C-trap have? So C-trap can be used to study nucleic acid protein interactions, or it can be used to, for example, study protein folding and conformational changes. In fact, we have a talk coming up next week on the use of C-trap for studying protein folding and conformational changes. If you're interested, it's next Tuesday. You can go to our website and register for this webinar. But today we want to talk about the applications of C-trap in studying cytoskeletal filaments and their motors. First, I will give you some examples of how C-trap can be used to study the activity of motor proteins, and how it can be used, for example, to study mechanical properties of cytoskeletal filaments. C-trap can be used to study the motility of motor proteins. For example, we can do the famous trip-bead assay with C-trap, where you have an actin filament tethered between two beads, and there is a third bead on a glass slide which is functionalized, which is coated with myosin. And then with the help of our high resolution, like the high resolution nanostage that C-trap has, we can bring the bead in contact with the actin filament. And Using confocal scanning, we can see that the actin filament is tethered between the two beads. One thing I want to mention here is that with the help of the microfluidics and the flow chamber, it's very easy to assemble an assay, like a complex assay like that, which usually takes a while to really get to a point where you can take your data. For example, it takes only 10 seconds to have an actin filament tethered between the beads and going on with the rest of your experiment. The way you do it is that, as I showed, like in the first channel, you will have the beads flowing, and once the beads are trapped, you go to the second channel where actin is flowing, and when actin binds to one of the beads, you move to the third channel where there is only buffer, and now you can move this bead toward the actin filament to form the tether of actin between these two beads. And once you have it, you can go to the fourth channel where you have these pedestal beads on the glass slide, and you can continue with your experiment. And if you want to do this in different buffer conditions, for example, different ATP concentrations or salt concentrations, you can, for example, move the beads and the tethered actin between them to the fifth channel where you have another buffer condition here. So once the myosin motor interacts with the actin filament and it generates the translocating force, it moves the actin filament. And using C-trap, we can measure this displacement of actin filaments, and we can measure the force that these motor proteins are generating. So the high temporal resolution of a C-trap makes it possible to measure this, the small displacements that these motor proteins are making. C-trap can also be used to study mechanical properties of cytoskeletal filaments. For example, this group has used C-trap to study the mechanical property, like the mechanical response of vimentin, which is an intermediate filament with a complex structure with these intrinsic regions, like the domains that have a different mechanical response, and they have used C-trap to study the mechanical response of these filaments. First, by fluorescently labeling these filaments, they use the confocal scanning to see that the filament is formed between the two beads. Now, by moving one of the beads and extending the filament, they look at the mechanical response of it by looking at the force extension curves. And as you can see here, using a C-trap, they've been able to apply high forces of like 600, 700 piconewton, which without going up to these high forces, they wouldn't have been able to get a full picture of the mechanical response of these filaments. Also, C-trap has made it possible for them that, because you can adjust how fast you want to move the beads, like how fast you want to extend the filament. They've done their experiment at different loading rates to see the effect of loading rate on the mechanical response of these filaments. C-trap has also been used to, for example, study the effect of tension on actin filaments in binding of actin binding proteins. For example, this group has used C-trap to tether an actin filament, which is fluorescently labeled between two beads that are functionalized with streptavidin. Then they put tension on actin filament, and they see how would it affect binding of this actin binding protein alpha catenin, which is also fluorescently labeled. So what they do is that at the same time that they apply force to the filament, they also take fluorescence images of actin and their actin binding protein. So at the beginning, when they start to apply force, they see that the actin filament is getting extended until it gets to its resting length. And at this point, the actin filament starts to feel the resisting force, and the force increases, and at some point the filament breaks. Now, in order to correlate how this tension in the filament is correlated with binding of the actin binding protein, they look at the ratio of intensity of the protein to actin filament over time, and they see that in the regions that there was tension on actin filament, the binding of the protein has increased. So this is a very nice way of combining force measurement and fluorescence measurements to see how tension in actin filaments can regulate binding of an actin binding protein. So as I said, so far I talked about optical tweezers combined with the confocal microscopy, but now we want to talk about the surface assay kit. So for experiments that needs to be done on the surface, optical tweezers can be combined with our surface assay kit, which is turf, wide field, and IRM. Since IRM is a less common technique, I'm sure that most of you know about turf and wide field. I introduced IRM more. So IRM is interference reflection microscopy, which is a label-free imaging technique, which is based on the scattering of light from the sample. And how can it be used? For example, we can have microtubules on a glass slide without any fluorescent label, and you can look at the growth and shrinkage of these microtubules, and you can look at it over the time to get information about the rate of growth and shrinkage of these filaments. Or for example, you can combine IRM with fluorescent microscopy. You can have microtubules without label on the glass and use IRM to image that, and have the proteins that bind to microtubules fluorescently labeled, and use a turf or wide field to image those proteins. And for example, we can measure the step sizes that kinesin motors make when they walk on a microtubule. We can have microtubules on a glass slide, and using IRM, we can image those microtubules. And now, because we know where they are on the glass slide, we can bring a trapped bead that is coated with the kinesin motor close to the microtubule, and by keeping the force constant, we can measure the 8 nanometer steps that kinesin makes on the microtubule. Again, the high temporal resolution of C-trap and the fact that we can keep the force constant makes it possible to measure these small steps of kinesin motors. So now that we talked about motors and filaments, I want to move to another section where we want to see how we can use C-trap to study the cell mechanobiology. So for example, we can use a trapped bead to apply force to the membrane of the cell to see how does the cell respond to that. For example, this group have used C-trap and they fluorescently labeled their receptors on the membrane of the cell, and they also biotinylated these receptors, and then they bring a trapped bead that is coated with, that is functionalized with streptavidin close to that cell. As you can see here, they bring the bead close and then they start to move the bead that way to apply force and deform the cell. And as they do it, they see that the receptors also move toward the deformed part of the cell. Can look at it one more time. So using these fluorescent markers, they can also track basically where the receptors are going. So now, because they're doing both fluorescent measurements and force measurements, so they can see that at the beginning there is when the bead is just in contact, there is not much force, but once they start to pull on the cell surface, they see that the force increases to 200, 300 piconewton, and they see this deformation in the shape of the cell, and they see that at the same time these receptors are accumulating in the deformed region of the cell. So C-trap can also be used to study the forces that filopodia exerts. So filopodias are these cellular protrusions that some of the cells have to communicate with their environment. And for example, we can bring a trapped bead near this cell to see how much force these protrusions exert. As you can see here in this movie, so when the cell feels like there is a bead in its environment, it extends these protrusions and moves toward this bead. So here in these snapshots, you can see better. So the cells feels the presence of the bead, and it has these protrusions that extend toward the bead. And now with the help of optical trapping, they can measure like how much force these filopodia are exerting on the bead, and by looking at the fluorescence images, they can see like the dynamics of these processes. Since here they have both actin and myosin fluorescently labeled. So now I want to spend a couple of slides just introducing, talk about introducing LUMICKS, that who we are. So LUMICKS goal is to improve human health by unlocking single molecule studies. And we do that by providing an instrument that makes single molecule studies accessible to researchers from different backgrounds. And we also try to make sure that our customers are successful, and we do that by helping them at different stages of their work, from experimental design to sample preparation and data collection and data analysis. This is a recent picture of our team. LUMICKS is a company that is based in Amsterdam, and we also have an office in Boston and in California. And finally, I would like to thank all our customers around the world that provided this data, and they shared their data with us. And finally, if you have any question, please feel free to put it in the question and answer section. And if you have more questions, feel free to contact me or Jason. And don't forget about our upcoming talks and webinars. Yeah, so I think that's for me, Jason.
Okay, thank you, Shima. Thanks, Shima, for the wonderful talk. Now we will start our Q&A session. So I'll read out the listed question accordingly, and our panelists will then address them respectively. But before everyone is busy and typing the question, I would like to advertise again that we are having we have actually quite many webinar upcoming. The most close one is next Tuesday. This will be a seminar focused on how to use C-Trap to do protein unfolding. We also have our keynote speak series. So the next one, the most close one, was starting from May 6th, Wednesday. This will provide by Dr. Ariel Kaplan, technician. He is a professor from Israel Institute of Technology. He will be addressed transcription factor binding and the regulation of gene expression. We also have another guest speaker who will present May 30th, also Wednesday, Dr. Ingrid Tesmer from University of Worcester. And her topic will correlate with DNA damage. Okay. So let's start for the first question. So thanks the question from John Bohr. So John Bohr asked about, so what's the software that the C-Trap use? Is that a command line based software or what is it? And how easily people can be trained for using that?
Yes, for calibration of C-Trap, basically we use the Brownian motion of the beads, and we look at the power spectrum, and these are all incorporated in the software. So basically, like you just need to trap a bead, and the software takes care of it. But it's based on the Brownian motion of the bead and fitting a Lorentzian function to the power spectrum. And yes, C-Trap can have like multiple traps. It has 2 traps, or it can have, for example, 4 traps if you have assays that you think that might need 4 traps. And right now it doesn't have time sharing, but you can do both fluorescence and force measurements as long as like your traps are like within a distance of 1 micrometer and not closer than 1 micrometer. I think, and about the forces, as I mentioned in one of the slides, like how do we measure forces? So we have a position sensitive detector that measures the displacements that the bead makes from the center of the trap. And because we calibrate the trap at the beginning of the experiment, so we have the information about the stiffness, and then the force can be calculated from that. I hope these have answered these questions.
Thank you, Shima. Frederick, we hope this answered your question. If you have follow-up questions, please feel free to type it again. We will answer them. So the previous one, Shima, so John Boy asked about the software. Could you tell us a little bit about the software that C-Trap use?
The software that's C-Trap uses, like basically controls different parts of the experiment. You can use it to control the microfluidic system. So you turn on and off the flow, like you, and for example, you can see your flow chamber, and you can, using a bright field, you can see your beads, like your trapped beads, basically in the software. And also it collects all the information throughout the experiment. As soon as you turn on the instrument, it keeps all the information, like all the displacement, forces, everything. When did you turn on and off the valve? So all the information is also like recorded, and then you can select which part of that data you want to basically use and proceed with that.
I see. All right. Thank you, Shima. So Fadri had a follow-up question. Did you mention 600 piconewton in one of the groups that use C-traps? Which laser power do they use?
Yeah, indeed. I said that they use the 600 piconewton. So with C-trap, you can apply forces from 0.1 piconewton to 1,000 piconewton. And this all depends on the laser power that you're using, and also like the size of the bead. So all of those affects how much force you can apply. I don't know exactly like what like how much was their laser power, but I can definitely send you the link of that paper that you can look at it. But I can tell you that the maximum power that C-trap, the laser output, is like 10 watts. But the fact that how much light gets to the objective, that's something else. But definitely I can check that how much power they were using.
Thank you, Shima. Fadri, again, if you have some follow-up, please feel free to type in. All right, cool. Glad we see that we answer your question. Thank you. Thanks for asking us the question. Okay, so while other people are busy typing, so I would like to introduce or say something about LUMICKS. So as Shima mentioned earlier, so LUMICKS is a biotech company. Our headquarters is at the Netherlands, Amsterdam, but now LUMICKS also had an office at Boston and California. So for people that in the United States, so we have now office application scientists, account manager, field application engineer that help us now to do the training and talk to individuals now who would like to learn more. And we, from time to time, go to different universities to give seminars. Now we try to broadcast now the single molecule tool, and hopefully this tool will also advance your research. Also, LUMICKS is a company, so we also have the Europe team and also the team in APR, Asian Pacific region. So if you are interested and you are in other regions, we do have people from other regions that could help you. Okay, I guess if there's no further question, lastly, so let me re-advertise again that there's upcoming webinar. The most close one is next Tuesday, focusing on protein unfolding using C-trap. Registration could be found at the same website that everyone registered before. If you have any question regarding C-trap, please feel free to reach out to us. Shima and I would love to help you, or you can also send your question or the info that you look for through the info@lumicks.com. Okay, so then I guess thank you for all your time, and we wish you have a great of the rest of the day. Thank you. Bye-bye.